So moving on to bacterial shapes, arrangements, and sizes. So they vary in shape, size, and arrangement, but they're typically described as one of three basic shapes. Coccyx, which would be round, sphericals, right? Bacillus, which are rods, and Coccobacillus are very short and plump. Vibrio are gently curved, and then Spirillum, and these are helical, twisted rod, or a spirochete, which is spring-like. To me, it looks like spiral macaroni. Um, so here would be a picture of a coccyx. So you can see that they're nice and spherical. The rod or bacillus, so you can see they look like a rod, right? Vibrio with a slight curve in it. The spirillum or spirochetes, right? They look like spiral macaroni. The spirochete is more curved. So spirillum is not as tightly wound as the spirochete, okay? We also can have branching filaments. They branch off, right? So you can see that there's like forks in the road. We also looking at, oh, that's what those pictures were. Sorry. So you can see the different microorganisms and what they were looking at, what these were, right? So F would be the Streptomyces species at 1000x. So you can go back and see what they are. We also have pleomorphism, and this is where they can vary in cell shape and size within a single species. And some species are noted for their pleomorphism. And this would be one example. We can have different granules, we can have rods, and you can have them be different shapes. Okay. So the arrangements of cells is dependent on the pattern of division and how cells remain attached after they're divided. So the cocci, these are singles or diplococci. Diplo is di, meaning two, so they'd be in pairs. Tetrads would be groups of four. There are also irregular clusters, chains, and cuboidal or cubical packets, which would be like Sarkinia. Your bacilli can either be diplobacilli, chains, or palisades. And so looking again at the different shapes and how we can arrange them, division in one plane, then you can have diplococci, which would be two cells, or streptococci, which would be multiple in a chain. The tetrad would be 4, or Sarkinia would be 8 to 64. So it just depends on how many there are. Look for the root words. Again, tetra is 4, so that would be, your tetra. Then you can have it in like clusters here, like staphylococci and micrococci. And then this is just showing the different sizes. So they can be incredibly small. So this is a human hair just to show you. And then you can see that these are very microorganisms, hence microbiology. So how do we classify our prokaryotes? Well, we're going to use microscopic morphology or shape, macroscopic morphology, which would be how they look in a colony, bacterial physiology, as well as serological analyses and genetic and molecular analyses. So bacterial taxonomy is based on Bergey's manual, and this manual of determinative bacteriology. There are 5 volume resources that cover all known prokaryotes, and they're classified based on genetic information, and this is called phylogeny or phylogenetic. And there are 2 domains, archaea and bacteria, and there are 5 major subgroups within 25 or with 25 different phyla. So if you look in your book, right, and we look at Bergey's classification, you can see that there are the volume 1 is the domain archaea, right? These are different ones that go in there, and I mean this is going to be in your book too, or you can refer back to it. So table 4.3 contains the slide with unidentified thermophilic, meaning heat-loving cranocoites, which are isolated from deep sea thermal vents and at very high temperature, right? So just the different classifications. And I'm just going to cruise through these because you can read about this in the book, right? And this looks like it's table 4.3 pretty much summarized, right? So the Bergey's is like the gold standard to figure out how to classify different microorganisms, though. And we're going to get some practice in this as we go through our slides and learn different stains and the different shapes and practice on the slides in the microscope in lab. Okay. So, some of these you'll say this one is the spirochete that causes syphilis. And, the grammata is the view of the budding cell through the fluorescent microscope. Okay. And the blue is going to be that nucleoid. So you can see it's very, very helpful on how to classify these different guys. Okay. So the major taxonomic groups of prokaryotes, again, would be the domain of archaea. And these are primitive and they're adapted to extreme habitats and modes of nutrition. And then there's the domain of bacteria. And these would be the phylum Protobacteria, and these would be gram-negative cell walls. Phylum Firmicutes, and these are mainly gram-positive with low gram-positive content. And we have the actinobacteria, and these are gram-positive with a high GC content. Okay, so here is your universal phylogenetic tree. So you can see your archaea, your bacteria, your eukaryotic cells, your prokaryotic cells, right? So you can see that's kind of just the universal organization. So why do we care? Okay, well, we use these diagnostic schemes for medicine. And so it's going to use phenotypic qualities in identification. In identification. And this is restricted to bacterial disease agents. And this divides bacteria based on cell wall structure, shape, arrangement, and phylogenetic traits. I'm trying to figure out why I can't see my recording button. Anyway, and then medically important bacteria. This would again be a table. So important family is the genre of the pathogenic bacteria with notes on some diseases. So the bacteria with gram-positive cell wall structure would be like the cocci, and these would be in clusters or packets that are aerobic or facilitative. And this would be in your family Staphylococcus. So these would be like your staph stains. Okay. Your cocci in pairs and chains are facultative, and this would again be in your family Streptococci, and this would be Streptococcus, right, with strep throats and dental problems. You have your anaerobic cocci in pairs, tetrads, which are 4 or irregular clusters, and these are involved in wound infections. You have spore-forming rods, like your family Bacilli, which would be anthrax, tetanus, gangrene, botulism. You have your non-spore-forming rods, and these would be in the Lactobacillus family. So this would be your food infections and other problems like that. You have acne, you have diphtheria, you have leprosy, and these are in these different families. And so you can see that this is important why we're looking at these different guys. These would be fun to make into your slide deck, and I'm sure that actually this is already in there. And so Nocardia, this is for your lung abscesses. Actinomyces, these are your dental infections. Streptomyces, this is a very important source of antibiotics. So we are very thankful for these, right? Then you have bacteria with gram-negative cell wall structures, and those would be like bacteria that are involved in gonorrhea and meningitis, Moraxella, right? And dental diseases. So your anaerobic cocci, your aerobic, so with air, coccobacilli, anaerobic without oxygen, right? You have your aerobic rods, and this would be pneumonia and burn infections. You also have miscellaneous rods, and this would be an undulant fever, whooping cough, Q fever, Legionnaire's disease, etc. You have facilitative rods and vibrios, and this would be like your salmonella, typhoid fever, dysentery, plague, and then you have your vibrio, which is that shape, right? This would be involved in cholera and food infections, etc. Then you have Campylobacter, and this would be an enteris. This would be Helicobacter for your ulcers, meningitis, and bite infections, right? So again, all of these are in your book, and you're going to be making these in your slide deck. So I'm not spending a whole bunch of time on these because you can study in the table, right? Your aerobic wound and dental infections, Lyme disease, and kidney infections, syphilis, right? rickets or Rocky Mountain spotted fever, STIs, and trench fever, cat scratch disease, right? And these are obligate and facilitative intracellular bacteria. And then you have the mollicutes, and these would be involved in pneumonia and UTIs. Okay. You also have like species and subspecies of these, and so a species would be a collection of bacterial cells that share an overall similar pattern or traits, in contrast to other bacteria whose patterns differ significantly. You also have a strain or variety, and this would be a culture derived from a single parent that differs in structure or metabolism from the other cultures of that species. And you also have a type, and this is a subspecies that can show differences in antigenic makeup. So that would be a serotype or a serovar, and this would be also susceptibility to bacterial viruses such as like the phage type, or in pathogenicity, and this would be the pathotype. So, quick little checkup. Round bacterial cells growing in an irregular cluster would be best described as what? You guessed, Staphylococci. That would be fantastic. All right, good job. All right, and then one more video.